In this video, we're going to focus on the regulation of gene expression in prokaryotic cells. But before we get into the nitty-gritty details of molecular mechanism and how regulated gene expression occurs, I just want to spend a moment thinking about this issue from a larger perspective, from the context of regulation of cellular metabolism and cellular function. We've already talked about this a bit when we talked about enzymes and their function and their regulation. We talked about the fact that very frequently in cells, in all cells, we have homeostatic feedback regulation that occurs such that an enzymatic pathway can be regulated by, for example, end products of the pathway. We talked specifically about a pathway that led to the synthesis of specific amino acids, for example, tryptophan. And as we discussed before, if the cell has plenty of tryptophan, then there's no need to expend the energy to synthesize more tryptophan. And so we might see feedback regulation at the level of enzyme activity, in which the product of this pathway would feed back and inhibit the function of this early enzyme in the pathway, typically a rate-limiting enzyme, for example, through allosteric regulation by binding directly to that enzyme at a distal site and preventing it from binding to substrates, right? So we talked about that, and we talked about the logic of that from the perspective of conserving energy and maintaining homeostasis in metabolic pathways. But now what we want to do is recognize that we can actually regulate this enzymatic pathway even further upstream by regulating the expression of the genes that encode these enzymes that are catalyzing each step of the reaction. And so very frequently what we see is that the metabolism and cell signaling that's going on in a cell will actually yield proteins that can interact at the level of DNA and influence the rate at which gene expression is occurring. So for example, rather than influencing the behavior of this enzyme, actually blocking the synthesis of the enzymes of the pathway, and so never expending the energy to make those, right? So we can see both types of regulation, and we're going to be focusing on this transcriptional regulation in this video. The example that we're going to be looking at is the first gene expression regulation mechanism that was ever worked out by a couple of French scientists, Francois Jacob and Jacques Monod, and they shared the Nobel Prize in 1964 for their discoveries because they were so seminal to our understanding of how gene expression can be regulated. And many of the basic principles that they figured out in prokaryotic systems are very applicable to what we see happening in eukaryotic cells as well, as we will see when we talk about eukaryotic gene regulation. All right, so we're in bacteria. That's the model system that was used, and that's the gene expression regulation system that we're going to be investigating. And there's a very well-known phenomenon, well-known now, that Jacob actually figured out back in the early 1960s, in which if you are growing bacterial cultures in the presence of more than one sugar, let's say glucose, and some other sugar, lactose, mannose, arabinose, whatever. You see this very characteristic growth curve that's referred to as dioxy, this biphasic growth curve, where we see, if we're monitoring the growth of the culture over time and determining the number of bacterial cells, or in this case, in this graph, we're looking at percentage of bacteria in the culture, we see this biphasic curve where we see an exponential increase in bacterial growth and then a leveling off or stationary phase where no further expansion of the population occurs, followed by a second exponential increase in the population and then a second stationary phase and leveling off. And if we followed this out even further as the nutrients were depleted in the culture, we'd see this culture population kind of crashing out and cells dying. Right? But this two-phase biphasic growth curve is very characteristic of providing bacteria a preferred sugar, almost always glucose, and some other sugar that they are less able to efficiently extract energy from. So if this were, for example, lactose, which is depicted over here on the left, we would see exactly this same kind of curve. And what's going on here? Well, it turns out that what's happening here is that the bacteria are utilizing their preferred sugar, in this case glucose. Why preferred? Because they are able to get more energy via glycolysis from this single sugar than from this disaccharide lactose, which is made up of a galactose sugar linked to a glucose sugar. In order to utilize this for cellular energy to make ATP, they first have to cleave this linkage between the two, and following that, they then have to isomerize this galactose sugar into glucose so that it can be fed into the pathway. So there's a little bit of a delay. There's a little bit more of an energy expenditure required to utilize this sugar. And so in almost all scenarios, glucose is going to be the sugar that the bacteria are going to go for first. And so the bacteria will utilize the glucose for their cellular ATP needs and divide and divide and divide. And only when the glucose has been depleted in the cell will we see the bacteria shift their metabolism and start utilizing lactose as the next preferred energy source, or the next available energy source for cellular ATP needs. And this lag that we see between the two growth phases, it is actually when the bacteria are shifting their metabolism and activating expression of the genes that are required to metabolize lactose. So in this stationary phase that we see between the two growth curves, we see activation of lactose metabolism genes and their expression. And so what we want to understand is how that shift happens at this point when glucose runs out. How does the cell then ramp up expression of the genes that are going to encode the functional products that the cell needs in order to utilize lactose as a sugar? So we really want to be answering two basic questions in this video. And the number one question is, how does the cell know that lactose is present? Right? Because it doesn't want to turn on expression of these lactose metabolism genes if lactose isn't the second sugar, right? If it's arabinose or mannose or some other secondary sugar, then it would be ramping up production of the genes and gene products required to metabolize those sugars. So how does the cell know that it's lactose that's the second sugar? And secondly, how does the cell know that glucose has run out? Because as we see from the curve, the increase in gene expression associated with these lactose metabolizing genes doesn't occur until glucose runs out. So these two conditions both have to be met in order for us to see the activation of these genes associated with lactose metabolism. So we want to understand how that occurs. And what we're going to be looking at is the set of genes that encodes the proteins involved in lactose metabolism. And as we've talked about before, we know that clusters of functionally related and coordinately expressed genes are called operons. And so we're going to be looking at the lactose operon and how it's regulated to turn on the expression of those genes only when these two conditions have been met. Lactose is present and glucose is absent. All right, so if we zoom in to the, for example, E. coli, circular chromosome, its DNA, and look at the region of the genome where we would see these genes, the lac operon, here's what we would see. We would see three so-called structural genes that are called LAC-Z, LAC-Y, and LAC-A. So we call them structural genes because these actually encode protein, right? So these fluorescent, protein coding. These fluorescent green blocks of DNA here are protein coding genes. And what do they encode? Well, they encode different gene products. The LAC-Z gene encodes a beta-galactosidase enzyme. And this is the enzyme that's required to cleave and begin the metabolism of lactose. We'll look at that a little bit more in just a minute. The LAC-Y gene encodes a lactose permease, which is a membrane transport protein, and the LACA gene encodes a transacetylase, the function of which is really dispensable for lactose metabolism. We don't really understand the significance of it. You can knock out this LACA gene and really have no impact on the ability of E. coli to utilize lactose as a sugar source. So we're going to ignore the LACA gene. But key point here is that these structural genes encode proteins that the cell needs in order to be able to metabolize lactose. So here's our lactose. Remember, that's a galactose and a glucose linked together, so it's a disaccharide. Here's our beta-galactosidase enzyme, and this enzyme catalyzes the reaction that cleaves the bond between the two sugars, releasing individual galactose and glucose monomers to the cell. Now, importantly for this gene regulation system, beta-galactosidase also catalyzes a second reaction, what's an isomerization, using this sugar. And what it does is it isomerizes the link between the two monomers such that we no longer see the 1 and 4 carbons linked together by this glycosidic bond. Instead, what we see is the 1 carbon linked to the number 6 carbon on our glucose. So isomerization of the covalent bond between the two sugars. So this isomerization yields a isomer of lactose called allolactose, and we're going to see that this is actually a very important component of the gene regulatory system that we're discussing. Okay, so what is the permease doing? Well, as I said, the permease encodes a membrane transport protein, and it is the membrane transport protein that's responsible for actively transporting lactose from the medium outside the cell into the cell cytoplasm. And it turns out that this membrane transporter is a co-transporter or symporter. It's catalyzing actually active transport using a proton gradient that has previously been established. So we have a higher concentration of protons outside the cell than in, so we have a concentration gradient, and the permease transport protein utilizes that concentration gradient as an energy source, which then drives the transport of lactose against its concentration gradient into the cell, allowing it to really take up lots of lactose even when lactose concentrations start to build inside the cell. Okay, so those are our two critical components for lactose metabolism in E. coli. All right, so if we go back up to the lac operon, we see that in addition to these structural genes, and as we've talked about before, we're going to see a common transcription regulatory region that controls the expression of all three of these genes at once. As we've talked about before, when we see transcription of an operon, we see one long polycistronic mRNA formed, which contains the sequences for all of the protein coding regions of that operon. In this case, there are 3 protein coding genes, and so those are all present in our multi-message mRNA that's produced, and then each of these would be individually translated to give us our beta-galactosidase, permease, and transacetylase proteins, right? So we've talked about that before. All right, so let's take a look at this transcription control region in a little bit more detail. When we talked about this before, we really only talked about the fact that there's going to be a promoter sequence at the beginning of our gene to direct RNA polymerase to bind there and begin transcribing. But now that we're talking about regulation of gene expression, we need to think about other sequences that may be nearby or maybe at a distance that are going to regulate how RNA polymerase interacts with this promoter. So we're talking about gene regulation, the influence of the rate of transcription of a gene. So the influencing of the rate at which RNA polymerase is going to interact with this gene and transcribe it into mRNA. So in the lac operon, what we see, in addition to the promoter, are 2 adjacent regulatory sequences. So these are sequences of nucleotides. The first is called the operator, and the second is called the cap binding site. Cap is a protein, and this is the site at which cap binds. And we're going to talk about what cap is doing when it binds in just a minute. But we have these 2 regulatory sequences on either side of our operon promoter. And they're going to be influencing whether or how often RNA polymerase sits down here at this promoter and begins transcribing. So what are these sequences? Well, it turns out that the operator is a binding site for a regulatory protein that negatively regulates transcription of this operon. It's a repressor protein. So the operator is where repressor protein can bind, and when it binds, it blocks RNA polymerase from being able to transcribe this set of genes. That repressor protein is actually encoded by a separate gene in the genome, so it's not part of the operon. And this is called the LAC-I gene. It has its own promoter. It's not regulated. It's always expressed, and it's always going to be producing this repressor protein that will come and sit down and bind here to the operator sequence. And when it's bound, it's going to block RNA polymerase from binding to the promoter and being able to transcribe. Okay, so the cap binding site, as I said, is a binding site for cap protein. And we'll talk more about that in a minute. The operator sequence is the binding site for the repressor protein, which blocks transcription, and the promoter, as always, is the binding site for RNA polymerase. So how do these transcriptional regulatory elements control the interaction or regulate the interaction of RNA polymerase with this operon and therefore the transcription of this operon? So we're going to take this in two parts. Remember, we were trying to answer two questions. The first of which is, how does the cell know if lactose is present at all? And that sensing mechanism relies on the repressor protein. So the repressor protein is the molecular mechanism by which this operon senses the presence of lactose. So here's our I gene, slightly different depiction of the lac operon, same idea. Here's our I gene encoding our repressor protein, which is this little crescent-shaped disc here. So when there's no lactose around, this repressor protein binds to the operator sequence, as we've previously discussed. And when it's bound here, RNA polymerase is going to be blocked from getting past this repressor protein and transcribing the operon. But this regulatory protein, it turns out, is allosterically regulated by allolactose, which, as we said before, is an isomer of our lactose sugar. So allolactose allosterically inhibits DNA binding by the repressor protein. So just as we talked about when we were discussing enzymes previously in the class, when we have an allosteric inhibitor of an enzyme, it changes the shape of the enzyme so that it can no longer catalyze its chemical reaction. In this case, when allolactose binds to the repressor protein, it causes the repressor protein to change its shape and no longer be able to bind DNA. So it hops off the DNA. When repressor protein is not bound, then there's no blockage here preventing RNA polymerase from transcribing this set of genes. All right? So in the absence of lactose, the repressor protein binds. When lactose is present, lactose gets isomerized to allolactose, which binds to the repressor, allosterically inhibits its ability to bind DNA, and the repressor comes off, right? Potentially freeing up this operon to now be transcribed. So this is how the operon senses whether lactose is present or absent, by whether repressor protein is bound or not bound. All right, so how does the cell detect the presence of or absence of glucose? Well, this depends on that second protein that we were talking about, which is CAP, catabolite activator protein. This, in some textbooks, depending on if you have international versions, might also be referred to as cyclic AMP receptor protein. You'll see both usages. CAP is more common, but sometimes you will see this referred to as CRP. It's talking about the same thing. All right, so what we need to know about this operon and the promoter before we can talk about the function of CAP and cyclic AMP is that the promoter is, so get our orientation here. This is our operator sequence that the repressor binds to. We just talked about that. This is our promoter sequence, and this underneath our CAP protein is our CAP binding site. Remember that we talked about promoter sequences, and that promoters have specific sequences of nucleotides that can recruit in eukaryotes general transcription factors, but in prokaryotes RNA polymerase itself, to sit down and bind and begin transcribing. Well, if the sequence of nucleotides in the promoter doesn't match the ideal or consensus sequence of a promoter very well, then it's going to be very inefficient at recruiting RNA polymerase, and it's going to be what we would call a weak promoter. All right, a strong promoter very effectively recruits RNA polymerase to bind. A weak promoter does not. And so what happens with the lac operon is that even if repressor protein is not bound here because allolactose has pulled the repressor protein off by changing its shape, this promoter, being a weak promoter, is not going to very effectively recruit RNA polymerase to come and sit down and bind, right? RNA polymerase may come and bind and maybe zip off one little RNA transcript, but it's not going to do that very efficiently, and so we're going to see very weak expression of the operon unless we have cap protein bound to its binding site right next to the promoter. If cap is bound, then RNA polymerase is going to be recruited to come and bind here very tightly, and is going to be able to zip down this operon, transcribing abundant lactose operon mRNA, and we're going to get lots and lots of protein synthesis from that larger concentration of mRNA in the cell. All right, so RNA polymerase is stabilized on this weak promoter by binding of the cap protein right next door. It turns out that cap protein can only bind to DNA when cyclic AMP is bound to it. All right, so cyclic AMP is an allosteric activator of cap binding. So only when cyclic AMP is bound to cap do we see cap binding to its binding site, and only then do we see RNA polymerase stabilized on the promoter and lots of transcription of the lactose operon. Okay, so we think about this logically. If cap is the mechanism by which the operon knows if glucose is present or absent, when does it make logical sense that we would see cap binding here? When glucose is there or when glucose is not there? Well, logically it makes sense that we're going to see this binding when glucose is not there, and that's exactly what happens. When glucose is being actively transported into the bacterial cell, what we see is a decrease in cyclic AMP concentration. Why? Because it turns out that the proteins and enzymes that are associated with transporting glucose into the cell and phosphorylating it so it stays there blocks the activity of adenylyl cyclase, which is an enzyme that we talked about before in our cell signaling unit. We recall that adenylyl cyclase is the enzyme that synthesizes cyclic AMP from ATP. Does the same thing in bacteria as it does in our cells. So if glucose is being transported into the cell at high levels, this enzyme is going to be blocked in its activity, right, inhibited, and therefore concentrations of cyclic AMP are going to be low. If concentrations of cyclic AMP are low, cap is not going to be binding next to RNA polymerase, and we're going to see weak promoter binding and transcription of the operon, right? However, if glucose is not being transported into the cell, then it won't be inhibiting adenylyl cyclase. We will see lots of cyclic AMP being produced, and that will bind to our cap protein, stabilize RNA polymerase on the promoter, and we'll see lots and lots of transcription, and following that, translation and synthesis of our permease protein and beta-galactosidase enzyme that's required to metabolize lactose. And in that way, we're now going to see a cell that has shifted its metabolism and its metabolic program to now be able to utilize this secondary sugar. Okay, so when glucose is present, once again, cyclic AMP is going to be low, no binding here next to the promoter in the cap binding site, and therefore no RNA polymerase stabilization, right? If lactose is also not present, we're going to see repressor protein bound here. And so we're going to see no transcription for two reasons, right? Now, what's the alternate scenario? Let's say that there's lactose present, but glucose is also present. Well, if lactose is present, we know that our repressor protein is going to be pulled off by our, not lactose itself, but allolactose, isomer of lactose that's going to bind and allosterically inhibit DNA binding by repressor. If glucose is present, we're going to see no cap binding, right, because it doesn't have cyclic AMP bound to it. So again, we're going to see very little mRNA being produced here. Why? Because of this weak promoter that doesn't very efficiently recruit RNA polymerase. It may sit down and, zip off one little mRNA transcript, but it's not going to do that very effectively. Only when we have this combination of environmental conditions in which lactose is present and glucose is absent do we see cap binding because it is being activated by cyclic AMP, no repressor binding, and very efficient transcription of the lactose operon. Now, what I've told you is true. I'm not telling you a lie. However, recent research seems to suggest that this regulatory mechanism is actually a minor component of the sensing of glucose and the metabolic shift to lactose or lack thereof in the cell. Because what seems to happen in addition is that transporting glucose into the cell seems to trigger some crosstalk that actually blocks lactose transport into the cell by the lactose permease. So in other words, as long as glucose is being transported into the cell, the transport of the alternate sugar is being inhibited, and so we don't actually see lactose accumulating enough in the cell to trigger synthesis at the operon. However, for other operons, for other sugars, CAP is the major regulator and sensing mechanism at the molecular level for glucose presence or absence. All right. So to make sure that you understand this gene regulation system, let's think about some different scenarios here and see if you can predict whether repressor protein would be bound or not and why, whether cap protein would be bound or not and why, and what effect that would have on the transcription and expression of these LAC-Z, LAC-Y, and LAC-A genes. Would they be highly expressed or not? So scenario number one, let's say there's no lactose around and there's no glucose around. Scenario number two, let's say that there's no lactose around, but there is glucose around. Scenario number three, let's say there's lactose present and glucose present. And scenario number four, lactose is present and glucose is absent. So go ahead and fill in the rest of this chart, making your prediction. Would we see repressor protein bound in this scenario if neither lactose is there nor glucose? Would we see cap protein bound, and what effect would that have on gene transcription? Do that for each of these scenarios, making sure that you are very clearly stating your rationale, and we'll talk about this during class when we do our gene regulation activities in a week's time.